And this is what affects tertiary structure. So remember that in structures of proteins, that primary structure is the actual sequence. Which amino acids are linked to which amino acids, linked to which amino acids. So like in this one, you have 8 amino acids linked by peptide bonds. That's the primary structure of the protein. Secondary structure is the helix and the beta-pleated sheets, the way that the backbone hydrogen bonds and creates folding. That's going to give some flexibility to the strand so that it's not just a straightened string. It's got a little bit of stretch to it. Hydrogen bonding of the protein backbone is what creates that secondary structure. So remember, the backbone is the strand along the top where you have nitrogens and carbons with double bonded oxygens, and so they create those twists and curves, and the hydrogen bonding creates that attraction that like stabilizes the structure. What you're looking at here is the R groups. So we went through with each of these R groups and designated, are they polar or are they non-polar? Because if they're polar, where are they going to be found? On the outside of the protein or deep on the inside? The outside, because they want to interact with water. So polar R groups, like the first and the second one, the fourth one, the sixth one, those
are R groups and side chains the same thing?
Yes. Okay, so the word R they came up with because it stands for residue. That's where the name came from. So side chain R group, it's the unique structure that makes one amino acid different from another. Okay, so the backbone is the same. So if you look, it's that NCC, NCC. That's the backbone that all amino acids have in common, and those are what get linked together into the chain. But the group hanging off of that middle carbon is the R group or side chain. And whether it's polar or non-polar is going to affect the twists and the folds that secondary structure creates. So this is tertiary. So tertiary, or the third level of structure, depends on the R group. Yep. So in tertiary, the R group, also called the side chain, either one, it is the part of the amino acid that's unique or different. So this R group, if it's polar, it is going to twist the protein so that the R groups will interact with water or interact with each other, because remember, like dissolves like. So ones that are polar are going to want to interact. Those that are non-polar are not going to interact with water. So polar groups will bend and fold to the outside of the protein, or they may cluster together as long as they're around other polar molecules. Non-polar groups, though, are not going to be on the outside. Non-polar is like oil, so the non-polar groups are going to go towards the interior. And so this is going to change from a helix or that beta-pleated sheet, and it's going to create extra folds, extra bends in the protein chain. Here's the example that I showed you last time, looks kind of like this. So you can see things. Here is non-polar. Remember, non-polar is hydrophobic, water-fearing. Over here, here's polar, hydrogen bonding between different R groups. You can even have polar groups that will want a hydrogen bond with water on the external. More hydrogen bonding shown down here. So you notice then you may have like that one area, it looked like it was an alpha helix, but because of the charged R groups, because of the hydrogen bonding, it caused that helix to like bend in half. And that structure is stabilized because of those R groups. So here's what they call a salt bridge. So a salt bridge is between a positively charged R group and a negatively charged R group. So that's going to create like an ionic attraction. So that's a pretty stable arrangement. The more of this, the more polar and nonpolar groups that you have, the more globular the molecule tends to be. The more folding you're going to get, the more three-dimensional shape that you'll have. Not all proteins have a lot of different variation in their R groups, and so some can actually remain in a more helical arrangement with less tertiary structure. And those they call fibrous proteins. Globular proteins have more tertiary structure. Globular proteins create more folds and bends. Fibrous proteins tend to have more helical arrangements. And fibrous, you think of things like fibers or strands or threads, and that's kind of the way that they look. So they give you examples of those in here. Globular proteins have more polar and nonpolar R groups, more folding. With globular proteins, the shape is important. So that arrangement, the shape, because of the polar and nonpolar interactions, is the important part, and a great example is an enzyme. So remember that enzymes interact with substances, and the reason they can interact is because of their shape. If they lose their shape, they lose their ability to interact. Receptors, so we'll talk about some of the functions, but there are some that have that shape that allows for their function. Fibrous proteins tend to look more stringy, and the two examples that they have here are keratin with the little sheep and collagen. So keratin, when you look at that structure, do you see that it looks like helixes? So pretty uniform looking, like the strands form this uniform arrangement. Notice that there's 3 of them, okay? So that's actually got more than one protein, but the protein's fibrous, and those proteins then wrap together. And that really looks like thread. If you actually take a strand of thread and you look at it really close, you can actually see that there's smaller threads wrapped around each other, and that's what adds the strength. So keratin, where do you find keratin? What's keratinized? Your fingernails, your hair, the epidermis, okay? So that's keratinized cells. It's these surface layers. So what's special about keratin? The reason that they show the sheep because of the hair. Okay, so that is a waterproofing protein. Keratin packs in and it tightens so that it excludes water. And so this is why if water gets on your skin, it like rolls right off. The only way for water to penetrate into layers of the skin is you use hot water and you have to soak for a long time. Because what happens when you sit in a tub for a long time? What happens to your hands? You get all pruned. Okay, so that is actually when water starts to penetrate into the outer layers and causes them to swell. But under normal conditions, that's slow to happen. That takes a good 15-20 minute hot water soak for that to happen. That's not going to happen just in a hand washing. So keratin acts as a waterproofing protein, and it's because it wraps tightly. These fibrous proteins can wrap tight and exclude water. The other one, though, looks a whole lot like it, but you notice that it's not as tightly packed. This is collagen. Collagen is the most abundant protein in the body. Collagen is the fiber made by most connective tissue cells. So when you think of connective tissue, that's not the epidermis, that's the dermis. That's the layer that holds your skin on. This is showing that yellow area, that's actually showing cartilage. So that is a knee showing the cartilage and shows that cushiony arrangement. Bone is collagen. The only thing that's different about bone, what gets added to bone? Calcium, and that's what makes the collagen hard. So all of your connective tissues are all collagen based. So this is really like a building block. Tendons and ligaments, what do? Tendons anchor, bone and muscle. So like think of your Achilles tendon. That's that tendon when you like bend your fingers back. You can see those sort of cords that stick out on the back of your hand, those are tendons. And those are what they look like microscopically. There's millions of them all wrapped together to make those tendinous cords. That's why I always think of them as stringy. They're good anchors. Special things about collagen, it is the anchoring protein. And it really looks like a string. So you can see how it can like tighten or anchor this thing to this thing. Think of tendons and ligaments. Ligaments anchor bones to bones. But this is the most abundant protein in the body. It is the protein primarily made by all of your connective tissue. So that's your loose, your dense connective tissue, your cartilage, bone, not blood, but everything else. Fibrous proteins tend to be stringy. Fibrous proteins tend to be insoluble in water. They don't dissolve in water, which is why they're better as ropes or anchors. Fibrous proteins are also tougher, okay? So their arrangement is more like a rope. They have some give because can you see how those alpha helixes would give you a little bit of stretch? So they're not going to snap easily. It requires a lot of force to tear tendons, to tear ligaments, to tear cartilage. Like those football players, you know, like they get clobbered and then they're all clutching and like they've pulled this, they've pulled that. It's always cartilage damage that ends up being suffering because of those high force injuries. But what about globular? Are globular is tough? If their shape is so important, no. Globular proteins, they're much more sensitive to changes in the environment. Where are you? Globular proteins can denature. They'll denature faster than fibrous proteins will because they have that tertiary structure. If you take the globular protein and you do any of these things to it, that will disrupt the tertiary structures. Globular proteins tend to be soluble in water, so you don't see them. But if you do these things to them, they unfold and they'll become solid. That unfolding, that loss of structure is called denaturation. And so examples of denaturation, heating. So what's an example of when you have something that's a liquid, you heat it, it becomes a solid? Like if you fry it. An egg, okay? Egg white is almost entirely protein. So egg white, and it's big proteins, but it's big globular proteins. Those albumins are large globular proteins. But when you heat them, the heat causes this folding to start to vibrate. Remember that heating, increasing temperature, changes the vibrational activity of the molecule. So being folded like this, it's soluble, but eventually it's going to lose that folding and unfold and it becomes solid. Can you go back? Will it go back to the gooey? No. Once you denature, that's a permanent thing. So it is difficult to actually refold it and make it soluble again once you lose solubility. Okay? So like cooking proteins, one of the reasons that you're worried about high fevers because high temperature, not necessarily going to fry, like in a true frying sense, but even heating above 105 degrees can cause denaturation of brain proteins. And that can end up leading to effects on whether or not brain function occurs. And that's why children, especially with those the little body volume, when their body temperatures go above 105, they really want to try to get that temperature down to try and avoid any kind of denaturation, especially with those developing neurons of developing brain structures. Second one, acids and bases. We did this in lab. So in lab with the lactase, where we did like half of the people added acid to their enzyme, the other half added base to their enzyme, and remember that it changed their activity. The reason it changes the activity is if you add hydrogen ions or you add hydroxide ions, that will change the ionic interactions. that can change the folded structure and can cause proteins to denature. Detergents. One of the reasons that you use soap is, what does soap have? Remember that it has those non-polar cores in the micelle. So that means that non-polar structures would get pulled into the soap molecules, and that's going to end up affecting the function. Changing the structure, changing the folding can end up affecting the function, can help in minimizing activity of any microbes and help to wash them away. What do we talk about with these? Silver, lead, and mercury. What's special about them, or what was the topic we covered with lead? So in lead, where did they find lead? Like, where was it used routinely before the 1940s? They put it in paint. Okay, so lead was added to paint. The reason they added is it would actually make the paint stick, and it was longer, like, lasting on a wall or on trim, even if it was outside. So, like, a lot of times you paint and like 5 years later it looks like you need to paint already, especially if you're like your house is like in the sun. It's much less heat resistant. They used to add lead because it actually helped the paint to stick to the wood, and they wouldn't have to repaint as often. The problem comes in is that lead paint does eventually flake off, and if you tasted lead-based paint, it would taste a little sweet. So this was a problem with kids living in homes that are 100 years old, because then it's pretty common that you end up with flaking paint, because 100-year-old homes don't have vinyl windows, the casements and everything is painted, typically was used just wood, and then those lead-based paints. So if you're a little kid and you find this little piece and you taste it and it's sweet, what are you going to do? You're going to continue eating it. Okay, so you're like, oh, this is pretty good. So it's not just like, oh, it's here, let me eat it. It actually does have a sweet taste. And so that's why they would continue to eat it. And the lead, though, builds up and affects, or it limits, development of proteins in the brain. So it ends up affecting brain development, and it's one that's harder to remove from the body once it's taken in. So that's always the challenge with the heavy metals. They're not excreted as easily. Once they get absorbed, they begin to collect. So it's also has to do with their size. So once they get absorbed, because you're talking about the silver, mercury, lead, they're big atoms, so they're not as easily filtered out. So that's why they banned lead-based paints. But if you live in a house that was built before the 1950s, then they have to do like a actual like disclosure when you buy a house to tell you if there was any lead-based paint used, and they actually have to go through to minimize exposure, not so much for adults, it's much more of a concern for children. Last one, mechanical agitation. So this is shaking. So when can you think of denaturation of proteins by shaking? Like, how do you do that? Like, what's an example of that? In cooking, there's two I can think of. If you whip eggs, what can you make? No. If you whip egg whites, what will it make? No. So whipped cream is a mechanical agitation, but whipped cream is when you take cream and you beat it on high, and that actually denatures the protein in the cream. But if you take egg whites and you whip it, it makes meringue. Okay? Have you ever had lemon meringue pie? That like kind of whitish stuff on the top? Okay? That's actually egg whites that have been beaten profusely, and it actually causes those proteins to denature. So mechanical agitation, shaking, whipping. Another reason, like the whole shaken baby, but usually that's like you worry about like brain bleeds and stuff like that when you shake little kids, so that's never a good plan. But those are examples: whipped cream, meringue made from egg whites. All examples of how you can just affect or change, especially tertiary structure. You can disrupt secondary structure, so you can damage, like you can cook, you can burn collagen with high temperature, but it's much more resistant. Globular proteins are the more sensitive because their shape is so important to their function. All right, so last one. So last one is called quaternary, Quaternary structure is the last level of structure. Quaternary structure describes how more than one protein together makes a large complex. So this, like collagen. Collagen is one helix, but then there is a second one that's also wrapped around it. And then there's a third, and all of these get twisted together to help make your ropey strand. The picture is better in the book, but that's my version. So 3 little helical coils all get wrapped together, twisted together, and that's going to make your tendons, ligaments, cartilage, and such. Their example of quaternary structure is hemoglobin. So hemoglobin's a globular protein. So can you see how it's got lots of folded nature? So this has definitely got polar and non-polar R groups to create this really folded structure. But it's not just one protein. Hemoglobin itself is actually 4. It's 4 proteins. 2 of them are colored in like purple and 2 of them are colored in gray. And you need all 4 proteins together to be able to carry oxygen. What are those things that are attached? So what is that? What is that piece? There's 4 of them. What does hemoglobin have in addition to just protein? It's what makes a red blood cell red. It's iron. So this is an iron atom. So each subunit has an iron contained, and that iron is responsible for binding oxygen. So when the red blood cell goes past the lungs, oxygen diffuses in and it binds to that iron. So iron, it exists as Fe plus 2, and then it goes to Fe plus 3. So it actually oxidizes. And so the oxidizing of the iron changes it from a real dark red to a really bright red. And this is the reason why oxygenated blood is bright red in color. But venous blood, deoxygenated blood, is what color? It's really, really dark red. It's really almost purple-black looking in color. It's all because of the oxygenation level of the hemoglobin based on changes in the iron. So quaternary structure, the last one, quaternary structure is when you have multiple proteins that are involved in this folding. They still may look fibrous or they may look globular with quaternary. But this leads us to the 8 big functions. So we talked about, when we talked about lipids, we talked about how lipids can be phospholipids, form membranes. They could be cholesterol and help in forming sex hormones. Protein's big function, there's really kind of 8 categories. And this first one, this first slide, actually shows all 3 in one picture. So proteins can be messengers, they can be receptors, and they can be transporters. So by messenger, it means that a protein can be the signal. It carries the information. The most common example of how proteins can be a messenger is with hormones. So hormones are proteins that are made by one cell, but they're sending a signal to another cell. So insulin is their example, one that we have talked about on multiple occasions in this class, because insulin's secreted by what? The pancreas. Secreted into the blood and it circulates. And it circulates until it leaves the blood, travels around cells, and it finds the second example. The second one is the receptor. So in this one, that green structure that's embedded in the membrane, that is the insulin receptor, And the shape of the receptor, globular, the shape of the receptor allows the insulin to fit into a pocket. And remember that means that insulin is that signal. It is to signal to the cell that there is plenty of glucose outside, that there is glucose in the blood. So notice in this one, outside of the cell, you have like six little glucose molecules. Inside of the cell, there's only 3. So high concentration, low concentration. When insulin binds to the receptor then signals to the channel, the glucose transporter. A channel in the cell opens up. And it doesn't open up for everything. It only opens up for glucose. So it allows glucose molecules to enter through the channel and pass through. Because remember, it's always going to move from high concentration to low concentration. And it's going to keep doing that until the concentrations are equal. As long as insulin is present, it binds to the receptor, and the receptor then tells that channel, open up and bring glucose into the cell. So there's 3: messengers, receptors, transporters. Then we have carriers. Hemoglobin. Hemoglobin is the carrier of oxygen. And it is the iron, which in this picture actually shows the iron as these little green pieces. So if you look, you can see 4 little green regions in the top and the bottom one. And you see that it looks a little different. The shape of the top and the bottom, a little bit different. The top one is hemoglobin without oxygen. The bottom one is what happens when it gains oxygen. So there's a little bit of a shape change that occurs as the hemoglobin is oxygenated. Remember I said that this is going to be the bright red? Oxygenated blood, sometimes called arterial blood, because it's bright red in color. And it actually causes that iron to become oxidized. When oxygen comes in, the iron goes from +2 to +3. It's oxidized. When it loses its oxygen, it changes slightly. The iron ends up losing the charge, going from a +3 back to a +2. It gets reduced again, but this turns into this very dark red, almost purple color. That is deoxygenated blood. So that's blood that you find in the veins. And that's really why your veins look dark in color, where they kind of look green under the skin. It's because it's very dark red blood that's contained within. Once it goes to the lungs, it reoxygenates, and this cycle just keeps on going. So carriers. So we have messengers, we have receptors, we have transporters, we have carriers. 5, protection. So there are proteins. The common example that they use is called an antibody. Antibodies are made by your immune system cells, and they're almost always drawn looking like a Y. So you always see them looking like this. And in fact, this side of the antibody is the important part. That side is the side that sticks to foreign substances. So if you have a bacteria cell, so a bacteria cell should have surface structures that are not like your cells. And if that's the case, then the antibodies begin to bind to it. And it's always like the ends of the Y that stick. When immune system cells start making antibodies, they can actually secrete millions of antibodies a second. So this becomes like a big production. But if you're a little bacteria cell and you're trying to be kind of sneaky, are you going to be as invisible if you suddenly have all this stuff sticking to you, collecting over the surface? No. And that actually helps to slow them down. It helps to keep them from dividing really fast, because they're not going to get as much nutrients because they have a big glob of antibody protein sticking to them. And antibodies actually mark the bacteria as foreign, and that will attract other white blood cells, especially white blood cells that do phagocytosis, that actually end up eating foreign structures. That's the reason that those antibodies are made. So they're going to help to attract cells that will eat the bacteria. They help to slow them down. They can neutralize toxins, really just kind of like keeping the bacteria from causing a lot of damage. Another example of protection are clotting proteins. So in your blood, you have proteins that are dissolved that you don't see. As long as the blood is moving, these are soluble proteins. The only time you see clotting proteins is when? If you cut yourself, okay? Or if the blood stops moving. So people can sometimes develop blood clots. That is because of poor circulation. If the blood stops moving because there's damage to the blood vessel, if there's jagged edges on the walls of the blood vessel, that will trigger clotting proteins. And what they do is one clotting protein sticks to a rough surface, and that attracts another, and that attracts another, and that attracts another. So they start to do this. So if you've got a cut blood vessel, this can help seal the hole. This can help stop bleeding. It's not such a great thing if that actually just starts happening randomly in a blood vessel, because you can see how it could cause a blockage. But clotting proteins are really designed to help stop blood loss. So they're also very much protective. 6 and 7, 6, contraction. So this happens where? What are the only cells that do this? Muscles. So in your muscles, and this is both skeletal, smooth, cardiac muscle. There are 2 proteins, and they're much more fibrous. And they look kind of like these bars, kind of like these long strands that are all wrapped together. Actin and myosin. Actin is the blue, the thinner ones. Myosin are the thicker ones. But what happens when the muscle contracts is this. So normally the actin and myosin slightly overlap. But when the muscle is stimulated, it does this. The overlap increases, and it increases at every layer. And so that, each one doesn't shorten but teeny tiny amounts, but you have billions of them along a muscle cell. So every single little overlap overlaps further, and that's going to cause the muscle to contract or shorten with variable amounts of force. Anchoring. We talked about anchoring, right? We talked about collagen. Collagen's the big anchor. Tendons and ligaments, good example. Another kind of interesting looking protein that is called elastin. And this is actually found in the connective tissue underneath the skin. And it is the reason that you can actually take and pull the skin up and it bounces back into place. Okay, so you go and pinch cheeks and it like bounces back. That's really good. Like the best elastin is when you're a baby. So babies have like little chubby cheeks, and if you squeeze them, they're still like nice and plump. But then if you do that to your grandma, what do you see? Not so much elastin. So what happens is the elastin has this real ruffled arrangement, and as you get older, the arrangement decreases. So they get less and less stretchy looking, and so you end up with wrinkles. Okay, so wrinkles are just where you have folds of the skin because of loss of that elasticity. So then you go get the collagen injections and everything gets all puffy. It's kind of interesting looking. But those ones are good for anchoring. The last one. And really the last one is what the last of this chapter is talks about, and that's the catalysts. So these are enzymes. So we talked about enzymes before. We've really covered it already. And remember that it is that tertiary structure. The globular arrangement of the protein is what allows it to act as a catalyst. Because enzymes do a couple of things. The things that it interacts with is called a substrate. Substrate, same thing as reactant. However, you might have one substrate or you could have 4 substrates. So whatever the reactants are, they just call it the substrate. Okay, so it's whatever your starting material that the enzyme's going to interact with. The enzyme, remember, brings reactants together in close proximity. So if you just had 2 things floating around, they would have to come together in order to react. Well, enzymes take this and push them together. Like the little E picture that I drew, how it pushes them together, same thing. So they can lower the activation energy by bringing substrates together. They do it in the right orientation. So I like this image because it shows, so if this is A and B can bounce against each other in lots of different ways. The only way A and B are going to be able to react is if they're just like this. And that's what the enzyme does. So it takes A and takes B and it puts them together in exactly the right orientation. So when you do that, can you see how this puts two molecules so close together that to make a bond is a very small stretch? So if they're far apart or if they're in this orientation, you're not going to have the two ends that need to be joined together. So the enzyme does this. So you can see it in this one. So in this, the right one is a glucose molecule. See how it looks like a little hexagon? So that's a sugar, the orangey looking one. And then over on that side, that's ATP. So that's an adenosine triphosphate. See the three little phosphates on it. But see how the orientation of these two pieces puts these very, very close together. So that the enzyme can just swap one of the phosphates over. And it's not a distance to be able to do it. So by putting them together in the right orientation, two bonds can easily be manipulated or swapped around, and that's going to make the reaction happen much faster. There's no way that we would survive if we didn't have enzymes, because reactions would happen too slow and you would not actually be able to adjust to changes in temperature, changes in nutrients, changes in demands from the environment. So how fast an enzyme works depends on a couple of things. So one is your raw materials. So if you don't have a lot of substrate, then the reaction is not going to happen very fast. And that's what this shows. So down here, if you have no substrate, no reaction. So if you add some substrate, the speed or the rate of the reaction goes up. And I like to think about enzymes as being, so they like take these two things, put them together, and then let them go. Take two things, put them together, and let them go. And they can do this, but they can only do it at a certain speed. So enzymes can't just speed up. They're going to go to a maximum speed. That maximum speed is up here. So they do have like a limitation. So they give you an example in here of like these guys. So these guys are our enzymes. And so they say, okay, if you have plenty of starting materials, plenty of parts, and the enzymes, the workers are in good shape, they can make this many bicycles in a day. I mean, there is a production number that you can do. And so you can't make more unless you add more enzyme. So you can have as many parts as you want. You could have 3 times the amount of parts. You can still only get 25 bicycles a day based on this, because the enzymes can only go at a certain speed. But pH can affect them because enzymes are globular. So what did we say? If you add acid or base, what can happen? It can cause it to denature. So if you change the pH, that can affect the function of an enzyme. It may make it work better, but it might make it look worse. And usually there is a pH optimum. So the optimum would be like, that's the best activity. So they show the guys, one guy didn't show up, one guy looks kind of rough. Okay, so are you going to make as many bicycles? Are you going to be able to do as much? No. And so this is because of, in this case, the example is this shows what happens if you have less than the optimal pH or conditions for the enzyme. Most enzymes run in this range. So the 6 to 8, that's very close to what? Very close to neutral. Most of your enzymes like pH close to neutral. Enzymes you find in the blood, they like to be about 7.4, because remember 7.35 to 7.45 is the blood's pH. So normally the pH is pretty close to whatever the environment. Why does pepsin like a pH of 2? Because of the acid in the stomach. So pepsin is designed to work best under acidic conditions. So if you had pepsin and you put it at a pH of 7, it's not going to work very well because its design is to have that acidic condition. Last one. So the amount of your starting material or substrate, the pH, and then temperature. So most enzymes also have an optimal temperature. Some people really like it at 80 degrees outside. Some people are like, this is the best weather ever. I'm not like that. I would actually prefer that it was 50 degrees year round. It's just me. Okay, I'm like, I can always put on more clothes, but like I can only get but so naked and like not get arrested. So it depends. Same thing with enzymes. Some enzymes like to work at certain temperatures. Other enzymes like to work at lower or higher temperatures. So here we have the hot and sweaty enzymes. And so you see again, they kind of look like they're struggling. And so what is their output going to be compared to their optimal temperature? Less. Okay, so again, below or above the optimal temperature, enzyme activity is going to drop off. Good example of this is temperatures that are associated with food spoilage. So most microbes that contaminate your food may have been present in your food to begin with. Like if you have like spinach or if you have anything that grows in the ground, even if you rinse, you're still not going to like sterilize that food. So most of your fruits and vegetables have some microbes, and those microbes live in the environment. So environmental microbes, they really like to live at 25 degrees Celsius, which is right about 75 Fahrenheit. So like room temperature, 70 to 75. So if you want to slow their enzymes, you want to slow their growth, what do? You put food where? Okay, so if you freeze it, then it's going to be kind of hard if you want to eat it. Okay, so if you want to have it so that it's still accessible, what would you do? Just put it in the fridge. Okay, so if you can drop it down a couple degrees above freezing, so you can drop 32 is freezing. If you can drop the temperature down to about 34, 35 degrees, it's not going to freeze, but it's going to slow those enzymes down. So refrigeration. So decreasing the temperature. And this was one thing that like, and I guess it's just because I'm a Yankee. When I first moved here, so we went for dinner at a friend's house, and like, so the grandma was like 80 years old and was like the big Sunday cook kind of person. So after lunch, where do they put the food? On the table in the dining room. So they like cover it and put it in the, and I was like, what are we doing? I was like, shouldn't we put all that in the refrigerator? And they're like, no, people just snack on it all afternoon long. And I was like, like that was really hard for me. So because like I'm used to, as soon as food is finished, you're done consuming, everything gets put into storage containers, everything goes into the fridge. And if you want it later, you'll warm it up. So that's like always been like the role, but that's like a really common practice. So to have like Sunday lunch and then later on, like in the afternoon, people like kind of snack on like the leftovers. And so I was like, okay. So refrigeration is just going to slow any environmental microbes down. So that's going to help to preserve food longer. Now, in doing any kind of food sanitation rating, what else do they look at? They go into refrigerators and they check temperatures. They take temperatures of cold foods. They take temperatures of all the freezers. What else do they check in temperature? Water temperature of where?
Like I know from my job, I go check the back sinks and stuff.
Okay.
The bathroom.
Okay, so make sure the temperature is not just cold enough, but it also gets hot enough. And if you have steam trays, which is where they put like prepared food that is to be served hot, the goal is food that's cold, you got to keep it cold. You have to keep it at 36 degrees or lower. So 36 to 34 is the temperature of cold foods. Hot foods, on the other hand, you don't want them at 25 because what's going to grow? At 25 is the optimal temperature for any microbes from the environment to grow. So that'll speed up spoilage. So instead, you heat them up. So you heat them up to 45 degrees, about 120, and that's also going to lower or limit. So hot food, like in a steam tray, hot food that is kept under a heat lamp, they increase the temperature, keep it above 25 because you see the same thing. Enzymes have slowed activity when they're not at their optimal temperature. Hospitals, and any dental hygiene, hospitals will use what's called an autoclave. An autoclave is really a pressure cooker. Okay, so if you've ever seen a pressure cooker, so pressure cookers are like a pot. You put your meat in, or you put your collards, they like to cook stuff like that. Stuff that takes a long time to cook. You put it in a pressure cooker with just a little bit of water. You close it, but it actually locks. The lid locks on, and then you begin to heat it. So the water boils and creates steam, and that steam heats. So the steam actually gets hotter than the boiling point of water. So water boils at 212 Fahrenheit. You can use a pressure cooker or an autoclave to heat steam to 240. So it's going to increase the temperature inside, and anything that's in there will be exposed to that higher temperature. So hospitals and dental offices, they use an autoclave to sterilize what? Sterilize instruments. So all of the stuff that they use to clean your teeth, yes, they wash them. Yes, they use a sanitizer when they wash them, but that's not all they do. They package them and they put them in an autoclave, which uses steam pressure to heat higher than the boiling point of water. So any microbes, they're not going to survive at that temperature. So when you pull out the packet, it's wrapped. So even if you touch it, you're not touching the equipment, but everything that's inside, any microbes are dead.
Is that similar to like that light they use to clean like the hospital rooms?
So that's ultraviolet light.
Yeah.
Okay, so you know, like if you go hang out in the sun too long, you can get a sunburn. Yeah. So those bluish glowy lights, those are actually ultraviolet lights. Okay. So ultraviolet light causes damage to cells. That's why you get a sunburn. But you use it as a permanent, like whenever an OR is not in use, it's like got this blue glow in the room, and as soon as people walk in, that shuts off. Because you don't want to be hanging out in there too long with the blue glow. You'd be all red face and peeling and everything. But that's what it does, is it actually damages any environmental microbes that might be on the floor, on the wall, on any equipment that's like stationary in the room, and helps to reduce it. So autoclaves can sterilize using steam. And so it'll go above 212 Fahrenheit. And that's a way to make sure that you're actually killing, not just inhibiting or slowing growth, but you're actually killing any microbes that are present. So the last topic is about inhibitors. So inhibitors will end up affecting how enzymes work. So they do just like what the word says. So they inhibit an enzyme. They're going to block an enzyme. Some of them block enzymes permanently. Some of them block enzymes, but they're irreversible versus reversible. So the first ones we're going to talk about are the ones that are reversible, which means they're not permanent. So there is competitive. So in looking at the picture, this is the normal, right? So normal enzyme binds the substrate, brings about the reaction, and releases the product. And then the enzyme can turn around and do this over and over and over again. So it just keeps doing this. So that's the normal enzyme function. But what's the inhibitor do? So the inhibitor is the blue piece. So if an inhibitor comes along, what happens? Where does it bind? It binds to the enzyme in the exact same spot that the substrate wants to bind. So this is our starting material, the substrate, and the inhibitor comes in, and as long as the inhibitor is there, can the substrate bind? No. So it actually stops or blocks. I think of it as being like a plug. So it's like the inhibitor is like a plug that sticks in the active site where the enzyme would interact with the substrate. And as long as it's there, the enzyme can't do anything. Okay, so it's a block. So as long as the inhibitor is there, the enzyme can't interact. So it's basically blocking its function. So we talked about this. Remember with wood alcohol. So wood alcohol was methanol. And methanol, if it's broken down by the liver, forms formaldehyde. And formaldehyde is preservative, is like embalming fluid. So if a patient came in and had accidentally ingested methanol, you would want to slow the breakdown of methanol. Because methanol could get filtered out by your kidneys, but you really want to slow this because the more formaldehyde you make, the more damage to cells, the more embalming fluid is made in the body. Same thing with antifreeze. So antifreeze works under a very same, that is ethylene glycol. And when it gets broken down, it forms oxalic acid, which is toxic and will cause permanent damage to liver and kidney structures. So you want to block that. Both of these are broken down by an enzyme in the liver. So what you can give them acts kind of as an inhibitor. So instead, you give them ethanol. The same enzyme breaks down all three of these. But when ethanol is broken down, it forms acetaldehyde. So it makes a person drunk. But it's not going to form into something that's toxic. It's not going to make embalming fluid. It's not going to cause the kidneys to shut down. So the ethanol, you administer this to block the other reactions from occurring. And so ethylene glycol, that green antifreeze, actually tastes sweet. And so if it spills on the ground because your radiator is leaking, then animals will actually come by and will ingest it because of its sweet taste. And so that's how you can have an accidental poisoning of ethylene glycol. So if an animal is brought into a vet's office, ethanol is what the vet will give them. So there typically is a bottle of vodka that is in the vet's office, and that's the administration. It actually acts as an inhibitor. So the ethanol takes over what the enzyme is. So the ethanol kind of takes the place of the ethylene glycol, and it gives time for your kidneys to filter the toxic substrates out. So both methanol and ethylene glycol are going to be toxic. So you'll give the patient ethanol as to give more time for the kidneys to get rid of the toxin, because it's used in exactly the same way. Now in this example, ethanol is broken down as well, but it still has an inhibiting effect on the reaction that forms those toxic products. Non-competitive inhibitors don't bind where the substrate binds. So if you look at this one, non-competitive inhibitors, they bind at a different spot. So there, down the left is the normal. Down the left is the enzyme normally binds the substrate. But on the right side, what does the inhibitor do? So the inhibitor comes in and binds at a different spot, but what does it do to the active site? Yeah, do you see the shape change? So for this substrate to bind, it needs to have like a pointy cleft. When the inhibitor binds, that changes it to a curve. And so now is the substrate going to be able to bind? No, because the shape changes. So a non-competitive inhibitor binds to the enzyme and changes the active site. And as long as that inhibitor is present, the active site isn't the same anymore. It's not going to work. So the enzyme just sort of sits there. It can't speed up this reaction. This happens with carbon monoxide poisoning. So remember we talked about combustion reactions. If you burn a fuel with not enough oxygen, carbon monoxide, CO, is one of the products that is made. Carbon monoxide binds hemoglobin. It doesn't sit in the exact same spot that oxygen does. It binds to the side of the hemoglobin proteins, and it causes a shape change. And the shape change means that oxygen can't bind. The problem with carbon monoxide is it actually binds really tight. So it clings to the hemoglobin better than oxygen does. So usually it's small amounts of carbon monoxide that's made, but small amounts and then small amounts continue to be made. Carbon monoxide levels build in the blood. Even though there's not a lot in the room, it just keeps on building up in the blood because it doesn't diffuse out. And so you end up having low levels of oxygen in the blood. As you increase CO, you'll decrease O2, and all of the effects end up coming into play, sort of the side effects. So then we have the last one. So the last one is the irreversible types. So irreversible means once the inhibitor binds to the enzyme's done. So it is going to bind permanently. So notice here, it looks like there is a connection. So that means that the inhibitor is not just sitting in the spot. The enzyme has now formed a permanent link to the active site. And so this enzyme can't work anymore. So this is permanently inactivated. And this is one of the problems that you have with silver, mercury, lead. So these ions will bind to proteins and not release them. And if they're bound to the protein, it ends up affecting the shape. And they're not going to release or move off. And so then you end up losing whatever function that protein had. All right, so the last one. So the last one is an example of an irreversible inhibitor. And it was one that was actually found on accident. So it's penicillin. So if you've ever taken Augmentin, if you've ever taken amoxicillin, if you've ever had penicillin, so those are all the same family of antibiotics, and they were actually found on accident. So Fleming was a researcher that was actually studying not penicillin, but he was actually studying bacteria. So bacteria, which is found all over, they are starting to isolate specific forms of bacteria, trying to study these single-celled organisms and how they live. So in order to grow them, you don't have to give them a lot of food. In fact, they grow them on what's called agar. And agar looks like Jell-O. It looks like a Jell-O jiggler, like it's in a little plate. It's sterile, but it's kind of a little bit mushy, but not really. If you press down hard, you can break through the surface. But bacteria, if you're going to grow the same sample, you'll spread this bacteria on the surface of the agar. In the agar is stuff like chicken broth. Just a little bit of salts, a little bit of nutrients. That's really all most bacteria need. Most bacteria are pretty hardy because, they live on the surface of the desk. They live on your skin. So they don't have like a huge nutrient requirement. So Fleming was studying this. And so one of the ways that they grow bacteria is they take bacteria and they just spread it on the surface of the plate. And they leave it, and it'll actually grow like a real uniform appearance on the surface. They call it a lawn because it looks kind of like uniform across its surface. So he spread these plates. He went off for the weekend, and he comes back, and he saw that there was mold on his plates. So he was like, because mold is like a nightmare, because mold likes to spread everywhere. So if you get it on a plate, you really have to just toss it and start all over again. As he tossed it in the trash, he pulled it back out and looked at it. And what he found was on this plate, so on this plate he had this lawn, except where he had this little mold culture. He had like this little glob of fuzzy, hairy, have you ever seen bread mold? Okay, so you know how it gets like, it looks like hairs? So he had this one little spot of hairy stuff. Everywhere else he had his bacteria, except right around where the mold was. So then he was like, well, why? Why didn't the bacteria grow right up to the mold? Why is there like this clear zone? What does the mold make that doesn't let the bacteria grow? What the mold makes is penicillin. So the mold, it's a natural product, so the mold actually secretes penicillin. so that the bacteria can't grow close to it and compete for nutrients. So what penicillin is, it's a irreversible inhibitor. And it actually blocks formation of the bacteria's cell wall. So most bacteria, and I'll draw a little E. coli. So E. coli is like a little rod, E. coli, salmonella, all those little rod shape. But they make a cover. So bacteria are a single-celled organism, and they make a nice thick cover to protect them from the environment. Because if they got to sit on a lab bench for a long time, they need to have protection from the environment. That cover is its cell wall. We don't have that, but this is going to protect this little single-celled organism. So cell wall structure reminds me a lot of chain link fence. So if you look at the bacteria cell wall, it is components, which are actually sugars, different kinds of sugars, that are arranged and then they're linked together. So there's links that are formed. And this becomes a real three-dimensional structure, so it's not just flat, so that's kind of like the framework of a building. So not only do you have it like this, but there's layers above and below, so you form this three-dimensional kind of cage structure. That's what that cell wall is. And that's going to inhibit stuff from being able to penetrate and damage the bacteria cell. So that's really protective. It also helps to kind of hide them, so it's harder for our immune systems to find it. But penicillin blocks the formation of the links. So you still start off with the little pieces. So you still start off. You still start off with all the pieces, but you can't make all the links. You end up with partial connections made. So what is this going to do? As a cell begins to grow, so little bacteria cells, just like your body cells, they have to grow and get bigger and then they can divide. So if you only have like a partial cell or partial cell wall and you grow and elongate, what will happen to your cover? If it's not solid and rigid, what do you think it can do? Does that look really strong? No, it'll actually grow right out of its cell wall. So if you then take that bacteria cell and now you've lost your cover. Because of penicillin, you can't make a good solid cell wall, so it breaks out of its cell wall. Now it's exposed. Now it can be damaged by the environment. Now it can dry out. Now it can end up having salt imbalances. Now it can end up being affected more by anything around it. So this is what fungus makes, and that's what kept the bacteria from being able to grow right up on top of it. It's an irreversible inhibitor in that it binds the enzymes that form the links. Bacteria are pretty smart though, because what do they make? What have we seen with antibiotics? What is developed? So have you ever taken penicillin and it doesn't work? Okay. There's antibiotic resistant bacteria out there. So what do antibiotic resistant bacteria do? They've changed, they have mutated, they have altered their function in order to not use the antibiotic. In fact, so there is MRSA, go back one, MRSA. Really? MRSA stands for methicillin-resistant Staphylococcus aureus. So it is one that is a huge issue in hospitals because it is methicillin resistant. So methicillin is a form of penicillin. So these bacteria can actually break or digest the penicillin So this is what penicillin looks like. So the methicillin actually breaks the penicillin. It makes an enzyme called penicillinase. It makes an enzyme that digests the antibiotic. And so is it going to have an effect? So you start taking it. If this bacteria can break down the antibiotic, then the antibiotic's not going to work at all. And that's the huge stress. That is the huge concern, is if you have a bacterial infection and it is a resistant bacteria, then you're not going to see improvement. You will steadily get worse because it's not going to have any of this effect on the growth cycle of the bacteria. That's an example of an irreversible. All right. So I guess you'll just have 9 and 10. So 9 and 10 for Thursday's test through this material. Make sure that you go back, use the exam review to help you in organizing it. I'll have this posted, so if you want to go back and look at any of the slides that are in here,